all participants. Today, topic for my presentation is culture media essential in clinical mycology laboratory. So in short, I will enumerate all the medias. So first, there are routinely used medias, and if we are suspecting any particular fungus, we will use special media for them. So first, I will discuss routinely used media. So in that, Accelerate Dextrose Agar with antibiotics without cyclohexamide. Then we have to again use Accelerate Dextrose Agar with antibiotic with cyclohexamide. Potato Dextrose Agar, cornmeal agar with or without Twin80, Dermatophyte test medium, brain heart infusion broth, blood agar and chocolate agar, Muller Hinton agar with 2% glucose. These are routinely used media. So first we will start with Accelerate Dextrose Agar. All the basics about this agar we all know. It is available in all textbooks. So now currently we are using AIIMS modification for SDA. So it is having 2% dextrose and neutral pH, that is 6.9. The third variety of SDA is SDA with 15% NaCl. So this media we will use for identifying some adventitious fungi. So we have to check the tolerance of NaCl. So if the organism is intolerant, then the colony diameter will be less than 2 at 21 days, and if tolerant organism, colony diameter will be more than 2. And fourth is Accelerate Dextrose Agar, Accelerate Dextrose Broth. So this SDA we will use antibiotics. So antibiotics we commonly use are chloramphenicol 50 mg and gentamicin 20 mg. So this is SDA with antibiotics. We use cyclohexamide. These are used for inhibiting the growth of some saprophytic fungi. But we have to use this media cautiously. In this, the cyclohexamide we are using for inhibiting the saprophytic fungi, but sometimes it will inhibit the growth of some pathogenic fungi like Cryptococcus species, Aspergillus, Lomentospora prolificans, Talaromyces magnifi, some Candida species, and some Mucorales. So if we are suspecting these fungi, we should not use the SDA with cyclohexamide. So here this is a picture. Here you can see these SDA with yeast colonies, and here with the Trichophytons terrestre. This is the obverse surface. On reverse there will be a pigment. Next is commonly used media is potato dextrose agar. It will support the growth of most of the clinically encountered fungi. So it can be used as a primary medium also. So this is most commonly used for slide culture for the stimulation and production of the conidia and pigment. So it is also suitable for maintaining stock cultures. And in the dermatophytes, differentiation of typical varieties of dermatophytes on the basis of pigment production will be very much appreciated on PDA. So these are some growths on PDA. This is Aspergillus flavus, and this is Penicillium trisogenum on PDA. So it can be used as a medium for primary isolation also. So and it is most commonly used for the slide culture procedure because it induces the sporulation. Next is a cornmeal agar. It is used with and without Twin80. So this cornmeal agar is used for the Dalma plate culture method for chlamydospore production in the yeast cell. So in the Dalma plate culture method, we will use cornmeal agar, and with that we will identify different species of the yeast colonies and Candida. All species of Candida can be seen. So if we add the Twin80, the chlamydospore formation will be good. So here this is the picture of the Dalma plate, and when after 48 hours we will see the growth under the 40x, if we will see, these chlamydospores have been, can be seen. It is the suggestive of Candida albicans species. So next is the rice starch agar. It is also a nutritionally deficient media and used for chlamydospore formation enhancement. So it produces chlamydospores faster as compared to cornmeal agar. But routinely we will use cornmeal only. This is less used agar. So next is mycocell agar. It is used for the primary isolation of fungi, like SDA. It is used to test an organism's ability to grow in presence of cycloheximide. So in this agar, compulsorily cycloheximide will be there, and some antibiotics will be there. It is a primary medium for isolation of fungi. So this is Brainart infusion agar here. So actually, it can be used for primary isolation of fastidious pathogenic fungi like dimorphic fungus. So and also for in vitro conversion of mycelial to yeast form, and vice versa can be done on the Brainart infusion agar. So for the better growth, we can add some blood or for prevention of contamination, antibiotics can be added further. So this was about BHI. So there is one media which will combine both the ingredients of SDA and BHI agar. So that is SABHI agar. It can also be used for primary isolation of fungi. So now I will be discussing the media particular to the fungus. So media used for isolation of Candida and other yeast. So for Candida we will use SDA with antibiotics without cycloheximide, because cycloheximide may inhibit growth of some Candida species. That's why without cycloheximide SDA we should use. Then blood agar, Brainart infusion agar, broth. Brainart infusion agar also, and we can use biphasic medium also in the blood culture. Then cornmeal agar with trinity, rice starch agar, chrome agar, Candida medium. These mediums can be used for the growth and primary culture. We can use another biochemical test media also. So these are fermentation broth for yeast, assimilation medium for yeast, rapid assimilation of Trehalose broth, esculin agar, and Christensen's urease medium. So I will be covering one by one. First, chromogenic Candida medium. So this is used for isolation and differentiation of yeast. It can be used as a sole primary fungal medium if specimens suspected of yeast predominantly. So it is based on the direct detection of specific enzyme activities by adding certain substances of fluorochromes to the media. So different species will give different type of color in this chromogenic medium. It can be used as a primary fungal medium for yeast suspected specimens. Also, it can be used for the detecting mixed yeast population. Like here you can see this in our lab only. When we have sticked on the chrome agar, two types of colors have come. One is purple, one is blue. So this is a mixed culture. So if there is any doubt that the Candida only we are having it's a mixed, then we can subculture it on the high chrome agar and check for the purity. So next is a biochemical test, fermentation broth for the yeast. So all the yeast species, Candida species can be identified by fermentation and assimilation. So fermentation tests can be done for the 6 sugars: glucose, maltose, sucrose, lactose, galactose, trehalose, cellobiose. These are the 7 sugars for identification of yeast. Here actually bromothymol blue indicator we will use, but we can use Andritz indicator also. So here we have used Andritz indicator, and if there is a gas production here inside now, we should add the Durham's tubes also. So that if there is a gas production, then it indicates the fermentation, and if it changes the color from pink to yellow, then it will indicate the acid production, which will indicate carbohydrate assimilation. So even if only gas is produced, then the sugar is fermented. Like this we should read. And if acid production is there, carbohydrate assimilation is also there. So all carbohydrates will also be assimilated, but not vice versa. So there are some assimilation media for the yeast. So there are 4 methods. Wickerham's broth method, in fact, carbon assimilation and nitrogen assimilation method mediums are there. Then Auxanographic plate method, this we are using in our laboratory for carbon assimilation test. This is most commonly used, and for nitrogen assimilation test medium. So third one is Agar with indicator method. That is for carbon assimilation test, and modified potassium assimilation method or test. So I will be discussing about Auxanographic plate method for carbohydrate assimilation test. So in this method, what we will do is like this plate. We will this is the carbon assimilation test plate, and on that we will put the sugar discs. So how to prepare that plate? First, you should prepare yeast nitrogen agar base 20 ml in a tube, which will be solidified. So whenever we are performing the test, we have to melt it into the boiling water bath. Then after we have to take the yeast nitrogen agar in molten form in the tube 20 ml. Add freshly prepared yeast culture suspension of 4 or 5 Macfarlane standard turbidity into molten yeast nitrogen agar. So whenever we are using, we have to do the melting of the nitrogen agar, and in that we have to add the freshly prepared yeast culture suspension of the yeast which we want to test. Then we have to mix thoroughly. Then after that we have to pour that yeast extract medium into the sterile 90 mm or 150 mm plate and allow to solidify. So this plate we have prepared with yeast nitrogen base and the suspension of the yeast to be tested. So and after it got solidified, we have to place the carbohydrate discs evenly placed on the plate and incubate at 30 degrees Celsius for 18 to 24 hours. And after 24 hours, we have to read, and any amount of growth around the disc indicates the assimilation. Like here, there is a growth around this disc, so this sugar is assimilated, and this sugar is not, because there is no growth around the disc. So next test, medium, that is a rapid assimilation of Trehalose broth. So this Arati broth will be used for the rapid identification of Candida glabrata. So actually, first we have to keep the yeast colonies ready. We have to select the yeast suspected of suspected of Candida glabrata carefully. The utilization of Trehalose in process of cyclohexamide is detected in this broth. So actually, this can be done by the this test will be done in the microtiter well. So 3 drops of Arati broth into microtiter well with controls we should put, and we have to add the colonies and incubate the plate for 1 hour at 37 degrees Celsius. So after 1 hour, we have to read the plate. If there is a color change to yellow, then it is positive, and it is negative if there is original blue or green color. So we have to put positive control as a Candida glabrata and negative control Candida albicans. So this was microtiter plate method. And we can do now rapid assimilation of Trehalose slide test also. So next is esquilin agar. This test is used for esquilin hydrolysis test is used for differentiation of Candida norvegensis from similar Candida species. So Candida norvegensis will be positive. Next is urease test, which is very commonly used in the laboratory. So it is used for the differentiation of yeast-like fungi and trichophyton species. So we have to incubate at 25 degrees Celsius for 7 days. Urease positive trichophyton is P. mentagrophytes. Negative, and if it is negative, it will be Irugram. So urease positive yeast are Cryptococcus species, Candida krusei, Candida apolytica, Trichosporan, and Rhodotorula. So now in 2021 only, there is a new medium came, Candida auris medium. This is a selective medium for isolation and detection of Candida auris, which is an emerging pathogen. It is this method is devised by Arunala Chakrabarti and team. So in this medium composition is actually yeast extract peptone, dextrose broth, sodium chloride in the concentration of 12.5%, ferrous sulfate solution, and agar. So it selectively grows only Candida auris colonies. So the specificity and sensitivity of the test medium were both 100% after 72 hours of incubation. This selective auris medium is inexpensive, can easily be prepared, and can be used as an alternative to molecular diagnostic tools in the clinical microbiology laboratory. So media used for Cryptococcus species, we have to use the SDA with antibiotics without cyclohexamide, because due to cyclohexamide, growth of Cryptococcus is inhibited. So we can use blood agar, chocolate agar. In the if we subculture the Cryptococcus colonies on the chocolate agar, the capsule production will be more, so more mucoid colonies will be. Capsule advancement will be there if we subculture on the chocolate agar. Then brain heart infusion agar, bird seed agar, sunflower seed agar. Then CGB agar we will use Canavanine, glycine, bromothymol, blue, CGB agar. Then next we will use creatinine, dextrose, bromothymol, blue, thymine, that is CDBT agar. And special media for pigment production are there, mustard seed agar and tobacco agar. So first we will see about the bird seed agar. So bird seed agar, this is for the primary isolation of the Cryptococcus species. It is a Niger seed. It contains Niger seed, and it gives the dark brown colonies or black colonies on this bird seed agar. So the limitation of this is dark brown pigment by Aerobasidium, Sporothrix, Hexaphylla, and Phyalophora can also be there. So other pathogenic fungi can also show the dark brown pigment, but Cryptococcus species primary media we will be use this bird seed agar. Similar to bird seed agar, sunflower seed agar is also there, that is pulse medium. This is also used for primary isolation of Cryptococcus species. Here you can see these are the pigmented brown color colonies. These are the Candida colonies. This is the these are Cryptococcus colonies. They're dark brown pigmented. Next we will see CGB agar, Canavanine, glycine, bromothymol, blue agar. This is used to differentiate Cryptococcus gatti from other species of Cryptococcus. The ability of Cryptococcus gatti isolates to grow in presence of L-canavanine and to assimilate glycine as a sole source of carbon is detected here. So here you can see this is cobalt blue color. The Cryptococcus gatti will give cobalt blue color, and other Cryptococcus species there will be no color change. So next is CDBT medium, that is creatinine, dextrose, bromothymol, blue, thymine agar. So this medium will be used to differentiate the Cryptococcus neoformans var groupi from var neoformans. So in this we will detect the thymine assimilation. So Cryptococcus neoformans var neoformans will give bright orange color after 5 days, and Cryptococcus neoformans var groupi will give no change in the color. These were the media used for the Cryptococcus. Now we will move on to media used for dermatophytes. So we have to use the SDA with antibiotics with cyclohexamide. Then dermatophyte test medium, dermatophyte identification medium, rapid sporulation medium, and polished rice medium. Dermatophyte test medium is used for the presumptive identification of dermatophytes, and it will give the red color due to liberation of alkaline metabolites. Here only dermatophyte will be grown, and if the dermatophyte has grown, then it will change the color from yellow to red. The medium will be red. This is a positive reaction here. This is a dermatophyte, and red color has been changed from yellow to red. So the growth, the color change will be there from 3 to 6 days only. So rapid growth will be there, and it should be incubated at 25 degrees Celsius. The disadvantage of this medium is it does not inhibit the saprophytic fungi, and true pathogens like Histoplasma capsulatum and Cystic dermatitis cannot be inhibited, are not inhibited. So they will also change in the produce a change in the color of the medium, and it cannot be used to study the pigment production. For pigment production, we have to subculture it on the SDO or beta or PDA. So next is dermatophyte identification medium. This is also can be used for the presumptive identification of dermatophytes. So the composition here in this indicator is bromocresol purple, but in dermatophyte test medium the indicator was phenol red. So so when the test it is positive, then from greenish blue to purple color it will change. When the dermatophyte will grow, the color of the medium will be purple color. So the advantage is this: it gives less false positive than the DTM. And here we have to incubate at 37 degrees Celsius, but dermatophyte test medium we have to incubate at 25. So this is the difference. So another medium is rapid sporulation medium for dermatophytes. It is similar to the dermatophyte testing medium. In this, we use the antibiotics chloramphenicol, chlortetracycline, and also cycloheximide. It promotes increased production of conidia, enabling better visualization of isolate color. So next is polished rice medium. This medium is used to differentiate Microsporum audioni from Microsporum canis. So actually in Microsporum audioni there will be no growth on polished rice medium, but canis will develop white mycelial growth with lemon yellow pigment. Now special media for cementitious fungi. In that 4 are there: casein agar, Lophoulus medium, tyrosine xanthine or hypoxanthine agar, and oatmeal agar. Casein agar is used for characterization of cementitious fungi, and it is given positive by Horte Wernicke. So Lophoulus medium is used for testing proteolytic activity of the Cladosporium species, but some saprophytic species can also give the proteolytic reaction. So the and some pathogenic species are non-proteolytic. So this is not a little bit non-specific. So next, special media for Malassezia species. There are 4 media: modified Dixon medium, Lemming-Notman medium, Datta and Dixit medium, modified Krumm-Agar, Candida medium. So this is modified Dixon agar. In this agar, characteristic polymorphology of the individual species is better demonstrated in the modified Dixon agar. So this can be used for the primary isolation of the Malassezia. Whenever the sample has come with suspected pityriasis versicolor fungus, causing fungus, then we have to go for this modified Dixon agar as a primary isolation medium. Then next is Lemming-Notman medium. This can also be used for primary isolation of Malassezia species, and it can grow all the species of Malassezia, and it provides a better recovery of some species, and it is good for the maintenance of Malassezia cultures. So next, third one is Datta and Dixit medium. It is a new this medium actually is used for the primary and rapid diagnosis of Malassezia, and for antifungal susceptibility testing of Malassezia, we can use this Datta and Dixit medium. So there is another medium, means modified Chromogenic candida for Malassezia species. In this, they have combined the components of Chromogenic candida with the composition of the modified L-1 medium, that is Lemming and Notman agar. The means contents, they are added to the contents of the Chromogenic, and with mixture they have devised the modified Chromogenic candida for Malassezia species. Here every species will give different color of Malassezia. Special media for Mucorales. For this we have to use SDA with antibiotics without cyclohexamide, because the Mucorales are inhibited by the cyclohexamide. We have to use the SDA with antibiotics without cyclohexamide. We can also use BHI broth. Then for sporulation induction, we can use Bhadade and Agelo technique, and soil extract agar by Mahapatra and water agar. So Bhadade and Agelo technique is used for inducing the sporulation of mucormycetes like Apophosomyces and Succinia species. It will be having 20 ml sterile distilled water supplemented with yeast extract solution. So on this agar, we have to incubate at 37 degrees Celsius for 7 to 10 days, and then check for the sporulation. So this is the paper which you can refer for this Bhadade and Agelo medium. The next, soil extract media. This is also used for inducing the sporulation of mucormycetes like Apophosomyces and Succinia species. And another use for this is it is used for mating H. capsulatum and B. dermatitis for the development of Clistothecea. So this is mainly used for the sporulation of mucormycetes. So next is water agar, which is a nutritionally deficient media, and it enhances the production of spores and conidia. It is used for inducing sporulation of Apophosomyces and Succinia species. So there are these are the special media for dimorphic fungi. Brain heart infusion agar, cystine heart and hemoglobin agar, yeast extract, phosphate agar with ammonia, alpha cell yeast extract agar, and soil extract agar. In these these media are used for yeast to mycelial phase conversion or vice versa. So now there are some azospore media, means Assimilation group fungi, they will produce the azospores. So those azospore, for checking the those azospores, we have to subculture them on the special azospore forming media. So these three media are available: acetate azospore agar, Gorodka agar medium, and V8 medium. Next is RPMI 1640 broth medium. So RPMI stands for the Roswell Park Memorial Institute 1640 broth medium. This is a completely synthetic medium, and it is supplemented with glutamine without bicarbonate with phenol red indicator. It is used for the broth microdilution method of CLSI M27A3. So and the last medium, Muller-Hinton agar with 2% glucose and 0.5 microgram per ml methylene blue dye. So this medium is used for disc diffusion method for AFST M44A2. In this, the glucose in Muller-Hinton agar, we have added glucose because it is suitable for the fungal growth, and methylene blue we have added to enhance the zone age definition. So Muller-Hinton agar is used because it produces acceptable batch-to-batch reproducibility, and the final pH should be 7.2 to 7.4. Thank you.